on YouTube, so we're going to start from here with a little introduction.
Okay.
So if you'd like to say who you are and where you're from and all that good stuff.
Yeah, that sounds great. So as I mentioned earlier, my name is Dr. Jeanette Metzger. I did my undergraduate here at UW and then also did my PhD thesis research here. My PhD is in cellular and molecular pathology, and specifically I studied Parkinson's disease. And my thesis research focused on what I'll be talking about today, modeling and visualizing Parkinson's disease in the heart. So after completing my PhD, I then became a postdoc and now a scientist in Dr. Marina Emborg's lab at the Wisconsin National Primate Research Center. And so the talk today will focus on using specifically a non-human primate model of the effects of Parkinson's disease on the heart. So Parkinson's disease is a very common neurodegenerative disorder. It occurs in about 1 in 100 people over the age of 60. And Parkinson's disease is commonly recognized by its cardinal motor symptoms, illustrated in the upper left here. So those 3 cardinal motor symptoms of Parkinson's disease are bradykinesia or slowness of movement, tremor, and rigidity. This tremor is often seen, as you can see here, in the hand, and the rigidity can be seen in back rigidity and stooped posture. And oftentimes we also see a shuffling or short gait in patients with Parkinson's disease. Now, these motor symptoms are associated with the main pathological findings of Parkinson's disease, which are those that are found in the brain. So this on the right hand of the screen here illustrates the main pathology that's found in Parkinson's disease, the damage to the brain. And so the main damage to the brain in Parkinson's disease is a loss of a group of cells in a part of the brain called the substantia nigra. The substantia nigra is very low in the brain, in the midbrain, and these cells, you can see here in a unaffected patient, they have this dark color to them, hence the word nigra and substantia nigra, so they're very easily visible even without any sort of staining or manipulating of the tissue. You can see that's in contrast to a patient with Parkinson's disease. So by the time a patient is diagnosed with Parkinson's disease, they've lost about 60 to 80% of these neurons in the substantia nigra. So the neurons in the substantia nigra produce dopamine, and there's dopamine used in a few different tracts in the brain and a few different functions in the brain. But these cells specifically send their dopamine on up to the motor system of the brain to help with movement. And so as these cells are lost, there's less control of movement in the body, hence the cardinal motor symptoms of Parkinson's disease. However, Parkinson's disease involves a lot of parts of the body outside of the brain, and this has been increasingly a focus of the research field and of clinical treatment of the disease. So this diagram here illustrates many different parts of the body that are affected in Parkinson's disease. So one extremely common symptom of Parkinson's disease is a loss of sense of smell. This actually often happens first before many of the other symptoms, and it happens in almost all patients with Parkinson's disease. In addition to that, we have things like reduced gastric emptying. So the time that it takes for food to leave your stomach takes longer. Associated with that also is constipation and poor mobility of the intestines. These are also very common in Parkinson's disease, about 80% of patients. And they can actually occur decades before the motor symptoms, as can anosmia, the loss of sense of smell. It can occur decades before the motor symptoms of the disease. We also have symptoms elsewhere. So I think I heard before I started talking today that there had been a previous speaker about swallowing difficulty in Parkinson's disease. So that's also extremely common and really affects quality of life for these patients. And the symptoms that I'll be talking about today are those affecting the heart. So the heart in Parkinson's disease has a loss of some of the neurons that innervate the heart. And these can lead to dysrhythmias, but also very commonly patients will experience problems maintaining their blood pressure and the stability of their blood pressure. So the heart changes its activity based on information from neurons that respond to any sort of stress. So this stress can be something like someone coming up behind you and dropping something really loudly and scaring you, or it can be stress that comes in the form of exercise or standing up quickly. Any of that is communicated to the heart through the sympathetic nervous system. So the autonomic nervous system that affects the heart includes the parasympathetic and sympathetic nervous system, illustrated here on the right. The parasympathetic nervous system is that rest and digest part of your nervous system that really focuses on digestion of food and sort of calming down activity throughout the body. The nervous system that's affected in Parkinson's disease is the sympathetic nervous system. So this is also known as the fight or flight nervous system. So again, this happens if someone comes up behind you and scares you, and all of a sudden you can feel your heart rate increase. You can feel your blood pressure go up. If you're just going from sitting to standing and you have to fight against the effects of gravity on your blood, you then need your cardiovascular system to work a little bit harder. And that's controlled all by this sympathetic innervation. However, in Parkinson's disease, the neurons that stimulate the heart to pump, these sympathetic fibers are lost. So this leads to a number of symptoms for these patients, particularly one I talked about earlier, orthostatic hypotension. So orthostatic is a change in position and hypotension is low blood pressure. So patients will often, when they're going from sitting down to standing up, report that they have a lot of dizziness and lightheadedness. You can also have fatigue that's made worse by this simply because your cardiovascular system isn't quite working hard enough. And as you can imagine, when you combine this with the motor symptoms of Parkinson's disease, this makes these patients particularly prone to falls, which can be quite dangerous and lead to a lot of hospitalizations for these patients. So this does have a very large impact on quality of life for the patients that suffer from the sympathetic nerve loss in the heart. And interestingly, like we talked about with some of the other non-motor symptoms of the disease, in many Parkinson's patients, the problems with the heart can actually occur before the motor symptoms will even start. And so patients won't know that these are contributing to Parkinson's disease often until they start to have the stereotypical resting tremor, often in the hand. But they might start to accumulate some of these non-motor symptoms up to decades before then.
Can it be diagnosed at that point?
That is an interesting question. Up until now, no. So up until now, these sort of combination of non-motor symptoms are not specific enough to know that what you're eventually going to have is Parkinson's disease. However, some of these non-motor symptoms and the stereotyped pathology behind them are getting more recognition as associated with Parkinson's disease. And so something I'm going to talk about here is our ability to see a loss of nerves in the heart. And within the last about 5 to 10 years, if you see a loss of nerves in the heart and you have some of these other start of the other motor symptoms, then the loss of nerves in the heart is now considered officially a supportive criterion for the diagnosis of Parkinson's disease. So it is recognized as a part of Parkinson's disease now, officially in the clinic, but themselves, they're not specific enough.
So you mean we can go in and diagnose the number of neurons in our heart and whether they're diminishing?
We can do imaging. Yeah, so I'll show you that here, actually. So this is a really good example. So this is an unaffected patient. And so this is the heart. It's upside down in this picture. So this is the left ventricle here. This is the right ventricle here. The left ventricle of the heart is the more muscular part of the heart that actually has to pump to the rest of the body. So you can see it's much thicker than the right ventricle. And what this is imaging in vivo in this patient for sympathetic innervation. So what we're looking at is where the sympathetic innervation is present in the muscular walls of the heart. And so the red is going to be this red hot uptake with lots of sympathetic innervation. So we can actually see that the sympathetic innervation, this fight or flight nerve fibers, are present within the muscle wall of the heart all throughout the left ventricle, but also that they're non-uniformly present. So we can see that there's more of them in some walls of the heart. There's also fewer of them down. This is actually the bottom of the heart, which cardiologists call the apex, and the top of the heart is called the base, just to be difficult. But there's less of them in the apex than in the base. Did you have a question?
How is that imaging done?
I'm going to talk about that in lots and lots of detail. Yeah, in about a slide or two, I think. Yeah. So this is positron emission tomography or a PET scan. And I will cover that, including which radioligand this is in just a second. But yes, an exciting question because that is what my talk focuses on today. I'm glad we're excited about PET scans. So if we compare this to a patient with Parkinson's disease, so this is one patient over a relatively short period of time. So we can see not only does this patient have a loss of sympathetic innervation in the heart, we can see that. Another important thing to see here is, again, it's non-uniform. Just like the innervation itself is non-uniform, where it is lost is non-uniform. So the loss is greater in the apex, and we can't see all of the walls of the heart here. We can only see the septal wall between the two ventricles and the lateral free wall on the side. If we could see all the walls of the heart, we would see that the loss is also greater at the back of the heart, the inferior side, that is the anterior. And then within a relatively short period of time, we have this very significant, appreciable, even just with this representative image, loss of sympathetic innervation in just a few years in this one patient. So this is a pathology that is progressive within patients.
And
So
what does the patient notice in that 2 years?
a worsening of their symptoms. So
Worsening?
the less sympathetic innervation you have in the heart, the less you can respond to that fight or flight need. So the more fatigue you'll have, the more difficulty regulating your blood pressure. So anything where you really need your heart to get up and get working is not going to be working as well with the less sympathetic innervation you have. So this brings me to a question that we already kind of got a sneaky answer to, which is how can we study the cause of this in potential treatments? So we'll talk about PET scans in a second, but first I wanted to talk about animal models, because of course today, while I do have some clinical images, I'm mostly going to be focusing, my work was in a non-human primate model. So animal models are really important to help us understand the causes and mechanisms of disease. I have a feeling that Jordana already talked about this in more detail than I will go into today. But animal models really allow us to get a more detailed snapshot of different phases of diseases as they are occurring, rather than waiting for things to present and be found in the clinic. And they also allow us to do some more investigative treatments and to see what actually might be able to intervene in the mechanisms as they're occurring. And also, the closer the model is to the disease, the higher the probability that the results of that therapy are actually going to transfer to the clinic. So as a really simplified illustration of what I mean by that, we have down at the bottom here examples of brains, because Parkinson's disease research focuses so much on the brain. Figured I'd bring the brains in at least once today. And you can see the difference here simply between the size of a mouse brain to a monkey brain to a human brain. There are also a large number of differences in the actual structures, and not just in the central nervous system, but also in the peripheral nervous system. So the rest of my talk today will focus on the peripheral nervous system, so I thought I'd discuss differences there too. So some differences between rodents and humans, and in this case, monkeys and humans have similarities. These are differences between rodents and monkeys and humans. The first is that the sympathetic and parasympathetic neuron cell bodies are actually found in different locations in the body, different locations along the spinal cord. These neuron cell bodies that provide sympathetic innervation exist kind of in clumps along the spinal cord. And because they're in different locations, we have a slightly different spread of where that innervation originates in rodents versus non-human primates and in humans. So that can make a difference in how we understand which parts of this innervation are affected by different types of impacts of the disease. And secondly, there are structural differences between the neurons themselves. So a good number of sympathetic nerve fibers in the heart are myelinated. Our group actually just published a paper on this like 2 days ago, which is really exciting, showing myelination in sympathetic fibers in monkey hearts, because typically peripheral sympathetic innervation is thought of as non-myelinated. So myelin is this protective wrapping around the axon of a neuron. So in this cartoon of a neuron, we have this cell body here. These are the dendrites that receive information from other neurons, a long axon that would then send information out to its neighbors. And this long axon in many neurons is myelinated with this protective coating, sort of like insulation around a wire, that allows for signal to travel faster and also provides some metabolic protection for this neuron. But in rodents, many of these nerve fibers in the peripheral nervous system are not myelinated. And so if we want to understand how these nerve fibers are damaged and how we can protect them, whether or not they have a protective coating can make a big difference in how we interpret the results of our study. And so things like this are what led us to want to do this research specifically in non-human primates. So PET scans, yay. So PET scans are positron emission tomography. So PET scans, it's a nuclear medicine, it's a functional imaging technique. It allows us to get a 3D picture of a functional process occurring in the body. So to do a PET scan, you will use a PET scanning device. This image here is of a micro-PET scanner. This is actually the exact micro-PET scanner that was used to generate all the images that I will show you from my thesis work from my part of the study. And you can't tell here because there's not really much to scale, but it's quite small. It is a micro-PET, not a human-sized PET. So we do have this smaller PET scanner that we're able to use that we have at the primate center, so we can do our scans there. And a PET scanner detects positrons. So in order for PET scanning to work, you need to use a radioligand. And a radioligand needs to have two main properties. First, it needs to have a positron emitting isotope on it. So this isotope will emit a positron, it bumps into an electron, and it creates two photons that are detected coincidentally by this detector and allows us to zoom in and see exactly where that radioligand is in the body. And the second thing that this radioligand needs to do is it needs to bind to, mimic, or accumulate in some sort of biologically relevant tissue. So we need it to go somewhere that we care about. And then once it's in that place that we care about, we need it to emit a signal to let us know where it is. So the image that we showed earlier was actually performed by PET scan, and we'll talk about it quite a bit more in a minute. So here, this is that image we looked at before. So as I mentioned, this is actually PET imaging in a human. This is not from our group. This is from a different research group that works more with clinical Parkinson's research. The PET radioligand that they use here is something called 18-fluorine fluorodopamine. So fluorodopamine looks a lot like dopamine. And so because it looks like a neurotransmitter. It's actually taken up by a neurotransmitter transporter that is on the sympathetic nerve terminals. And so it is taken up inside of sympathetic nerve fibers. It then emits its positrons there, and we can see exactly where all the sympathetic fibers are in the body. Specifically, in this case, we were focusing on the heart. So we can do something similar in monkeys. So this is kind of a busy image. The first thing to point out is that while we're looking at the heart up here in this image, down here the heart's right here. So this image is the heart on its side. This image you're sort of looking up from the feet of a monkey. And so this white hot spot right here is the liver. So the liver is going to take up a lot of our radioligands just because it processes a lot of things that are put into the body. And so unfortunately, in this case, the liver got in the way a little bit of this imaging. This was imaging that was performed by a previous graduate student in the lab, Dr. Valerie Jors. So it was her thesis research that actually created the non-human primate model that I'll be talking about today. And this is just a representative image from her work. And what it shows is that we can, first of all, we can see the heart in a non-human primate using a radio tracer. In this case, slightly different. This is carbon-11 metahydroxyephedrine. So this looks a lot like norepinephrine, but similarly, it's taken up by neurotransmitter transporters on sympathetic terminals. So it's taken up by these sympathetic fibers. So we were able to see the heart in our non-human primate using this PET radioligand. And not only that, but she then developed a model of a loss of sympathetic innervation in the heart. So to model the pathology that we see in Parkinson's disease. And we created that model by the administration at time point 0 here of a neurotoxin called 6-hydroxydopamine. So 6-hydroxydopamine is structurally very similar to dopamine. And so it's taken up inside of sympathetic nerve terminals. But once it's taken up into a cell, it then breaks down into toxic compounds, causing damage and killing the nerve fibers. And so you can see the effect of that here. So 1 week after we gave that neurotoxin, there was almost no uptake of metahydroxyephedrine. So in other words, almost all the sympathetic innervation was lost. We did have some recovery of the presence of sympathetic innervation in the heart from 1 week to 1 month. By 3 months, the lesion had really stabilized between these two time points. But you can see here what resulted is a loss of sympathetic innervation in the heart of this monkey that, similar to Parkinson's disease, was non-uniform. So not all parts of the heart were equally affected, but the toxin was very effective at creating this PD-like representation of a loss of sympathetic innervation in the heart, in this case, in a rhesus macaque. So once Valerie had created this model and we knew that we could model the pathology of this sympathetic innervation in the heart in Parkinson's disease, I then joined the lab. And so then we started asking questions about what we can learn from this model. So first we stepped back and said, okay, if we want to understand more about what causes the damage in the heart, let's think more broadly about what we know causes damage to neurons in other parts of the body in Parkinson's disease. And so more broadly in Parkinson's disease, there's two main neurodegenerative mechanisms that I'll talk about today. The first is oxidative damage. So this is a very simplified sort of cartoon style representation of oxidative damage. On the top here, this is really just illustrating that something we're all familiar with, which is cutting into an apple and it browns over time because you've now exposed the surface of that apple to oxygen. And oxygen is then able to react with compounds in that apple that lead to this browning and eventually some degradation of the apple. Well, similarly, inside of a cell, if we have an imbalance of reactive oxygen species and antioxidants, if we have too many reactive oxygen species inside the cell, those reactive oxygen species do just what their name implies. They go around and react with compounds in the cell, and they eventually cause damage and break those down. So oxidative stress over time is going to damage cells. And so we know from lots of studies in the brain that oxidative damage is a very common mechanism in the pathology in the brain of Parkinson's disease. Interestingly, we also have reason to suspect that this sort of oxidative damage would occur in the heart. And the main reason there is that in order to make norepinephrine, which is the main chemical transmitter for sympathetic nerve fibers, you have to go through a chemical process that creates lots of these reactive oxygen species. And so not only is this a mechanism involved elsewhere in Parkinson's disease, but we have reason to believe that it would specifically, that these sympathetic fibers in the heart would specifically be prone to oxidative damage. So a second neurodegenerative mechanism that we'll be looking at today is inflammation. So starting on the lower right here, this figure kind of takes us through an explanation of why we think inflammation is likely involved. So any sort of damage that happens to neurons in Parkinson's disease or from another disease If it's damage that eventually leads to death of these neurons, the immune system is going to get involved. Because once you have cells that are dying, you have to recruit the immune system to help clean everything up. But the problem is, as you recruit more and more inflammatory cells, if more and more neurons keep dying because of this ongoing, say, oxidative stress or other neurodegenerative mechanism, you're recruiting more and more immune cells, and eventually you get to a point where those inflammatory cells are actually themselves secreting compounds that lead to neuron damage. And they're recruiting too much inflammation, and you can lead to this self-perpetuating cycle of neurotoxicity. So as I talked about, many of these mechanisms of neurodegeneration have been studied fairly well in the brain, which then left us with our question. Now that we've developed our animal model and we have some idea of the mechanisms we want to look at, we want to know if we can image this ongoing inflammation and oxidative stress in our animal model to see if these imaging techniques could then be useful to teach us more about how the heart is affected in actual patients with Parkinson's disease. So this work was all published in 2018 in Nature Parkinson's Disease. So this is covering in vivo inflammation and oxidative stress markers in our non-human primate model. And the timeline that was used in this study is shown here. So that neurotoxin that I talked about that's able to cause a loss of sympathetic innervation in the heart. This is the same neurotoxin that was used by the previous researcher in our lab, 6-hydroxydopamine. That was given at this time point 0. 1 week after that, we then performed our PET imaging. To have something to compare that to, we also had performed PET imaging 3 months before we did anything to the animals. Well, a variable amount of time before we did anything to the animals, 3 months to 1 month, to get baseline PET scans. And then finally, as I showed earlier, Valerie's work demonstrated that our lesion had stabilized between about 1 and 3 months. And so then we wanted to look again at 3 months after the neurotoxin was administered to answer questions about whether or not the inflammation and oxidative stress were still present, and whether or not we had successfully modeled the sympathetic loss in the heart in Parkinson's disease. And so there were 5 adult male rhesus macaques that were part of this study. So first, I want to show some representative images of the marker of oxidative damage in the heart. So this is a PET radio tracer called copper 61 ATSM. And essentially this radio tracer is able to just enter inside of cells. And once it's inside of cells, it has this basket-like structure. And when it encounters areas with lots of reactive oxygen species, the oxygen species react with the basket-like structure, and the radioactive copper just pops right out, and it gets integrated into the cellular copper machinery. And so it just gets stuck there. And so wherever we have areas with lots of reactive oxygen species, you'll have an accumulation of this radioligand. And so you can see here that sort of like in the earlier study, the liver decided to get in the way a little bit. And so rather than having nice graphs of what everything looked like in the left ventricle, I have representative images. But you can see here that even at baseline, before anything was done to these animals, before we administered the neurotoxin to create the model, there is some amount of uptake of the radioligand in the left ventricle. 1 week after we gave the 6-hydroxydopamine to these monkeys, this same monkey, you can see this dramatic increase in oxidative stress in the heart. And then by 3 months, it had essentially returned to baseline values. So our PET scans are able to show us in vivo, in a living animal, a time course of the effects of oxidative stress in the left ventricle in these monkeys in this model of Parkinson's disease sympathetic denervation in the heart. So luckily, we were able to get a lot more detail for our marker of inflammation. And so once we had that more detail, we had to decide how to present it. And so I actually worked for quite a long time with Dr. Colleen Moore, who is an emeritus professor from the Department of Psychology at UW. And so she has a lot of experience with statistics, but also a passion for ecology, which came in handy here. Because what we decided to do here was display the uptake of this inflammation marker using a type of plot that's used a lot in ecology called a surface plot. So this type of plot, you can imagine someone walking around an area outside and saying that they count a certain number of bees or whatever they're doing their ecology study of, and then making this map of the intensity of where they found things in this sort of 2D area. We decided to adapt this a bit to show us what the inflammation was like in the heart. So you can sort of think of this map as analogous to the map of the world, and the left ventricle is analogous to a globe. So what we have moving across the bottom here, this x-axis, are the regions of the heart. So the septal region between the 2 ventricles, the anterior region at the front, the lateral region on the side, and the inferior region around the back of the heart. Moving on this y-axis, we have the different levels of the heart from the base to the apex. And then on this z-axis here, which also corresponds to the plot over here, the intensity and the height on the z-axis both represent the uptake of the radioligand. So wherever we have a higher point on the graph here, we have more uptake. So for example, here, looking at this graph, this is just at baseline. The number of immune cells in the heart were on average a bit higher up at the top, at the base, than they were down at the bottom. We also have a bump here in this anterior lateral region, which should be about here in this diagram. And so we do have some uptake, some presence of inflammatory cells in the heart just at baseline. At 1 week after we give the neurotoxin, again, a dramatic increase compared to baseline. And interestingly, as we've seen with sympathetic innervation, it's non-uniform. It's not the same throughout the entire left ventricle. The increase is much higher up at the top of the heart than at the bottom, though there is an increase throughout the entire left ventricle. And again, by baseline, we've returned essentially to baseline values. So this is an average from all 5 of the monkeys in this graph. So this brings us to the next question, which is whether or not we can interrupt these mechanisms. Now that we know that oxidative stress and inflammation are occurring in the heart, can we interrupt these mechanisms to actually decrease the damage to the nerves of the heart? Well, to test this, we looked at a drug called pioglitazone. So pioglitazone is actually FDA approved to treat type 2 diabetes. However, pioglitazone has also been shown in many different studies to be able to decrease inflammation and oxidative stress. So interestingly, in 2015, there were 2 studies that came out that made us want to investigate pioglitazone. The first is pioglitazone was investigated for whether or not it could slow down Parkinson's disease, and they found that it did not slow the progression of the symptoms. However, if you think back to the first slide where we talked about the damage done in Parkinson's disease, you don't have motor symptoms of the disease until you've lost 60 to 80% of those neurons in the substantia nigra. And so then the concern there is that potentially at this point, when we're trying to preserve innervation, that we might be intervening a little bit too late to have a significant effect on the progression of the motor symptoms. And the reason that we're suspicious of that is because that same year, a second study was published that followed diabetic patients that had taken glitazones. And they found that in the population of patients taking glitazones, there was a 28% decreased risk of diagnosis of Parkinson's disease. So this makes us ask whether or not early detection of Parkinson's disease, such as by imaging of the mechanisms and early intervention, could be important for something with this sort of a mechanism of action being effective for those patients. So our group has worked with pioglitazone in the past. This is a non-human primate model of the motor symptoms of Parkinson's disease. So this is actually that part of the brain, the substantia nigra. And these animals, to model the effects of Parkinson's disease, they get a different neurotoxin than what I used, a neurotoxin called MPTP. It's given intracarotid so that it only goes into the brain, and it actually only goes to one side of the brain. So here the toxin is affecting only this side of the brain. In the animals that received pioglitazone immediately following the toxin, there's significantly less loss. This is all the brown here is a marker for the neurons in this part of the brain. So significantly less loss with pioglitazone. And this was associated with decreased inflammation. So down here, we're looking at the same part of the brain, but we don't have a marker for sympathetic innervation. We have a marker for inflammatory cells. So the side of the brain that got the neurotoxin and no pioglitazone had a lot of inflammatory cells. The side of the brain that got the neurotoxin and pioglitazone had many fewer inflammatory cells associated with the preservation of the dopaminergic neurons in the nigra. So then the next question, we have our animal model. We have some ideas for how to image it, and we know that the animal model effectively showed increase in oxidative stress and inflammation. So we wanted to know then whether or not pioglitazone decreased these mechanisms in the heart. And so this timeline is actually, you can probably tell, the same timeline as before. So this part of the study was done in parallel with the earlier part. This is 5 additional adult male rhesus macaques. But instead of only receiving the 6-hydroxydopamine at time point 0, 24 hours after that, they began daily oral dosing of pioglitazone. And they had PET imaging at the same time points, 1 week after toxin, baseline before toxin, and 3 months after toxin. So these on the left here are the same graphs as before. This is that same representative animal. What we've added here is graphs from a pioglitazone, or images, representative images from a pioglitazone animal on the graph of the data. So one important thing to point out is that you can't actually compare brightness from side to side due to the way that the medical physicists collected and displayed these images. There's a different threshold for maximum white brightness between these images. So instead we have to pay attention to the numbers. So if we look at the numbers here, our marker of oxidative stress, as we saw before, in our placebo animal, it increased at 1 week and went back to baseline by 3 months. In the animal that was treated with pioglitazone, it also increased, but significantly less. And then again, back to baseline by 3 months after toxin. And we can see that graphically here. So this shows the mean and the standard error of the mean. With all 5 of the animals in each group here, we can see that in both of our groups from baseline to 1 week, there was some increase, but significantly greater in the animals that did not receive pioglitazone. In our placebo group animals, they had much more oxidative stress in the left ventricle than the animals that received the pioglitazone treatment. So again, our marker of inflammation. So again, these are those surface plots that sort of wrap around the left ventricle. And so similar to the placebo group, in the pioglitazone group, we can see that there's some uptake of this just naturally at baseline. There are some immune cells just naturally present in the heart in an animal at baseline. And again, it's non-uniform. So similar to the placebo group, a little bit more up at the top than at the bottom, and this bump in this anterior lateral region. If we compare the effects at 1 week, very clearly we can see that while there was an increase in the placebo group, there was no increase in the pioglitazone group. And again, at 12 weeks, while the placebo group resolved to baseline, the pioglitazone group, there were no statistically significant differences between these time points. So the inflammation that was caused by the neurotoxin was effectively stopped by the administration of pioglitazone. And finally, we want to know what actually happened to the sympathetic innervation in the hearts of these animals. So again, we have surface plots. We were able to get these high detailed view of the heart. So as we discussed in human patients, sympathetic innervation in just a naturally occurring monkey heart is non-uniform. There's a little bit more up at the top than at the bottom, a little bit more in the subtimal and anterior area than in the inferior, the back of the heart. And this is true in both our placebo group and our pioglitazone groups. These are effectively the same treatment group at this point because this is baseline. Nothing has been done to any of these animals yet. After the toxin, we have a very dramatic, almost complete loss of the uptake of this radioligand in the heart. So it's an 86% loss of uptake in this group and an 82% loss of uptake in this group. And then by 12 weeks, this is when things start to get really interesting. So I added the blue shapes here to point out areas that were statistically significantly different between these 2 treatment groups. And so what we saw is that by 12 weeks in both groups, there was some recovery of sympathetic innervation, just like Valerie was shown in her previous study. And that recovery was greater up at the top of the heart than at the bottom of the heart. And interestingly, there were specific areas in the heart where pioglitazone seemed to be more effective at preserving the sympathetic innervation in the heart. One of those areas was in this anterior region up at the base. You can see that the recovery is pretty good in the septal anterior region in this animal, and that extends over into the lateral region more when given pioglitazone. You can also see overall the recovery was just sort of greater up towards the base. And then interestingly, down at the bottom of the heart, in the apical region, we had this septal area that had a statistically significantly more recovery when given pioglitazone than the placebo. So in conclusion, the innervation of the heart is damaged in Parkinson's disease, and this can be modeled in non-human primates. This was the work by Dr. Valerie George back in 2012 that then sort of opened the door for me to be able to do my thesis work on the mechanisms of the loss. And PET imaging in our non-human primate model of PD cardiac damage successfully detects changes over time in cardiac inflammation and in oxidative stress. And in the non-human primate model of cardiac nerve loss, pioglitazone decreased both inflammation and oxidative stress and preserved nerves in the heart, but interestingly, in specific parts of the heart. We saw those 2 parts, one in the base and one on the apex earlier. So this makes me think of some next steps. So these PET radioligands could be tested in human patients as a next step to help determine the role of inflammation, oxidative stress, and Parkinson's disease cardiac nerve loss. And this gets us a little bit back to the conversation that we had much earlier in the talk, which is if we can potentially detect inflammation, oxidative stress before the nerves are damaged in the heart, if we can detect that there's something wrong, there's something ongoing in the heart before we have this massive amount of neurodegeneration, is there anything we can do to intervene? And if so, do we need to intervene quite early when we detect these, but before there's so much neurodegeneration that damage has already really affected the quality of life of that patient? Should we intervene early, and can it be with a treatment such as pioglitazone that targets some of the these cellular mechanisms, inflammation and oxidative stress, and the damage to the heart. So with that, I'd just like to thank the huge team that it took to do this research. So of course, in the Emborg lab, my PI, Dr. Marina Emborg, and other members of the Emborg lab, Dr. Vita Bondarenko, Henry, Helen, and Rachel were students that I worked with, members of the UW-Madison Cardiovascular Research Center. So I didn't show this in my talk, but in addition to doing PET scans, we also looked at the health of the heart via EKGs in these monkeys. We drew blood to look at circulating catecholamines. We did imaging to look at their activity. We did quite a few other things as well that others at, for example, the Primate Center helped with as well. And of course, the medical physics department. So these medical physicists, some of them actually made the radioligands that we used at the cyclotron, which I think is really cool. And some of the other medical physicists were there to help me interpret and gather and understand the data. Dr. Colleen Moore was hugely helpful when I was flooded with numbers and had no idea how to run statistics or anything. And Dr. Kathy Gallagher, she is a physician scientist. She's a physician with the VA and with UW. And because of this background with patients, she was able to make sure that we stayed sort of focused on answering clinically relevant questions and making sure that our model used imaging techniques and things that could potentially be translated and be clinically relevant to the patients that she sees. And then funding, of course. And at this point, I would be happy to take any questions that you have.
We'll put that up there. Who's on your committee?
Yeah, so I don't have my full committee up here. Kathy was on my committee. Dr. Raghu Ramaganti, who's in neurosurgery, was on my committee. So in addition to this work, we did some work looking at whether or not damage to the heart causes changes in blood levels of different RNAs. And so he specializes in that sort of work. Dr. Bo Liu was on my committee, and Jim Holden, who retired very recently from the medical physics department, Dr. James Holden. He was in medical physics for a long time. He was fantastic. Yeah, he also helped me wade through the massive amount of data. When you're not used to looking at PET scan data, it's a lot. It's a lot to try to understand. So I was really helpful to have a committee to help guide me through all of that.
So are human trials about to begin or what?
No, for this one, there aren't any human trials for the effects of pioglitazone on the heart. So I think that's for a few reasons. I think one is, first, we need to figure out exactly what the mechanisms of cardiac damage in the heart in humans are. Because as I mentioned, not very much is known about that. So I think a more appropriate next step would actually be to try some of these radioligands to understand more about the mechanisms of damage in the heart. But there's not quite as much emphasis on the damage in the heart and clinical trials for that as there is for some of the motor symptoms in Parkinson's disease. So many of the clinical trials are more focused on the motor symptoms. Like I mentioned, there was that clinical trial for pioglitazone for motor symptoms, but it's complicated to start a medication that treats mechanisms of neuron loss when you've already had 80% of the neurons lost. That's been an ongoing problem in the field, is how do we identify these patients earlier? Is it appropriate to identify them earlier before they have an official diagnosis? Is it appropriate to test a medication in a patient that doesn't already have an official diagnosis but is likely to develop a disease? So yeah, all complicated questions that I hear about from the clinicians, but I usually kind of live in my world of monkey research. So interesting to hear about, but difficult choices that I suppose I'm grateful that I don't have to make myself every day.
Along those lines, Karen Gunderson at Home Mass, is there a test for Parkinson's disease?
So essentially a diagnosis for Parkinson's disease involves going to a neurologist. So you'll go to a neurologist and really what they're looking for are the cardinal motor symptoms. Because as of right now, there's not a blood test to confirm it. There's not an imaging test to confirm it. It's sort of like Alzheimer's disease, where technically the diagnosis isn't formally confirmed until autopsy, because there are other things that can cause Parkinsonism. So Parkinsonism is the presence of all of those motor symptoms of Parkinson's disease. So for example, you can have Parkinsonism caused by a stroke in the part of the brain in the substantia nigra. So you can have a stroke that damages those cells, and it would cause all the same motor symptoms, but it would not be full Parkinson's disease. And so then you would be less likely to have some of these non-motor symptoms of the disease, because there's a different etiology with a stroke versus one thing I did not talk about today is there's actually a second pathological hallmark of Parkinson's disease. So in addition to the loss of neurons in the substantia nigra, you have an accumulation of a protein called alpha-synuclein. So at autopsy, a neuropathologist is looking not only for a loss of neurons in the substantia nigra, but they're looking for this protein accumulation that's called Lewy bodies. So Lewy bodies are clumps of alpha-synuclein that are all clumped together, and they'll actually remain in the brain even after the neurons die. And so if you look at the brain of a patient that had Parkinson's disease, the neurons will not be there, but you'll see these clumps of Lewy bodies where the neurons were. And so somehow we don't fully understand why alpha-synuclein begins clumping up in the brain of patients with Parkinson's disease. And that's something I didn't touch on here, but it also clumps up in some of the nerve fibers in the heart in Parkinson's disease. So this non-human primate model, one caveat of this model is that it does not cause this protein clumping. And that's because protein clumping takes a very long time, but instead the model that we chose is a very fast-acting neurotoxin. So there are some animal models of this protein clumping. For example, you can have mice that overproduce alpha-synuclein, and they'll just produce so much of it. Their genome has been altered so that they just produce lots of alpha-synuclein, and they'll produce so much of it that it'll start clumping up. Pros and cons to that model too, right? Because then you're working with mice, which are further away from humans. They don't have exactly the same pathology as Parkinson's disease. So it's interesting when trying to decide what treatments are most relevant to move into the clinic. We have to ask questions about which parts of the disease are we mimicking accurately. So yeah, long answer to the question, but.
Then you mentioned Lewy bodies. I've heard Lewy body disease. Is
Yes.
that to distinguish it from Parkinson's?
Yeah, Lewy body disease is separate. Lewy body disease, you also have Lewy bodies, but it does not cause the same symptoms as Parkinson's disease. Yeah, so there are a few different diseases with Lewy bodies. Interestingly, patients with Parkinson's disease can also have accumulation of amyloid beta. So that's the protein that accumulates in Alzheimer's. So in Alzheimer's, you get accumulation of amyloid beta and of a protein called tau. But amyloid beta tends to accumulate in most people as they get older. It's just the rate is much faster in Alzheimer's disease.
Right.
And interestingly, there's some overlap in what a neuropathologist will find in the brain between Parkinson's disease and Alzheimer's. Yeah, so there are multiple of these protein accumulation diseases, and depending on where the protein accumulates, how quickly it accumulates, and the ratio of which proteins are accumulating, you get very different presentation of the disease. And we still don't know why. We don't know why with some people you get more accumulation in a certain part of the body than others, or why A beta and not alpha-synuclein, why Alzheimer's disease and not Parkinson's disease. That's unclear. For a very small number of people, the answer is genetic. But it's a very small portion of Parkinson's disease cases that have a clear genetic component.
Wow.
And those patients tend to have a very young onset. So you can have patients with Parkinson's disease in their 30s and their 40s. And for those people, usually it's something in their DNA that makes that gene different. But for almost all Parkinson's disease patients, it's what we would call idiopathic. So we don't know why.
Yeah, if early detection leads to some therapies that can address the inflammation and oxidation in the heart, would it be realistic to expect that those same therapies might address inflammation and oxidation up in the brain?
Yeah, I think that's a really interesting question. And I think so. I do believe that, for example, that's why pioglitazone was so interesting for looking at the brain, is because we do have some of those shared mechanisms between the two diseases. And if we think about it, inflammation and oxidative stress affect lots of different diseases. And so this idea that pioglitazone could be something that helps diabetic patients, but also potentially helps Parkinson's patients, or maybe could help Alzheimer's patients, is a very compelling idea. I think that, unfortunately, as you can tell by the fact that it hasn't been FDA approved to treat these diseases, we haven't been able to figure out how to test it in a way that answers the question, given the assumption that it needs to happen earlier. I think that's always one and that's why animal models are something that our lab likes so much, is because we can ask these questions and we know exactly when we're causing damage. And so we can start, for example, some people have tested pioglitazone starting before they cause the damage, to sort of represent what if I took this for a long time, way before any damage was happening to the body. Or our group chose to start pioglitazone the day after we gave the neurotoxin, trying to interrupt very early. But how we get there is a very challenging question, especially because, as I mentioned, all the non-motor symptoms of Parkinson's disease are so general, right? This decrease in sense of smell, constipation, until you get the characteristic sort of pill rolling tremor, that hand tremor, it's really hard to diagnose.
That smell is interesting because it's the same as the COVID thing now.
Yeah.
But I never heard, my dad had Parkinson's disease, and I never heard of lack of smell.
It doesn't happen to everybody. It happens to almost all patients, but as with anything with neurodegenerative diseases, it's never 100%.
And I'm not sure, maybe he did.
Yeah. Yeah. And interestingly, some patients sort of either won't notice or it happens slowly enough that they don't think anything of it. So I was with Dr. Gallagher once, and I was with her shadowing patients, and a patient came in who that day got diagnosed with Parkinson's disease because his tremor had just started. And when she started asking him about some of these non-motor symptoms, he was like, oh yeah, I guess I do have that. Oh, I guess I have been experiencing that. But it's something that he had just attributed to getting older, or attributed to just not feeling well, you know, like being constipated, or his sense of smell wasn't as good, or he'd get lightheaded when he stood up. And the problem is that a lot of times that can simply just be attributed to something other than Parkinson's disease. And so they really are so non-specific. They're very common in Parkinson's disease, but they're also very common in lots of other diseases, or with something that's not Parkinson's disease, simply just with age, some people can have a decrease in sense of smell or changes in GI motility.
Because my dad was a farmer and he did a lot of herbicide spraying for many years.
Yeah.
We just connected those two things and assumed that's what he was getting worse from, was Parkinson's, which was caused by herbicide spraying, which he wasn't very careful with.
Yeah, unfortunately, herbicide sprays are a known risk factor. For
They're a
example,
what?
a known risk factor for Parkinson's disease. For example, I just saw an ad, I was visiting my in-laws last weekend and saw an ad on TV for lawyers that were asking whether or not you had Parkinson's disease and had ever been around paraquat.
Right.
So paraquat rotenone is another thing that's associated. As a matter of fact, paraquat and rotenone have been used in animals to study Parkinson's disease, like symptoms. So yeah, it's a known association. Exposure to various chemicals is a known association with development of Parkinson's disease.
But we don't know if it really affected the heart at all then, do we?
No, the only way to really know, outside of imaging, I think the most common thing you'll hear from Parkinson's disease patients is this orthostatic hypotension, because that really stands out, because you'll go from sitting down to standing up and all of a sudden you get super lightheaded. So that's something that Parkinson's disease patients, once someone will ask them, have you ever experienced this? They'll be, oh yes, this happens to me all the time.
Yeah, but who hasn't?
Yeah, I know. And that get back to the question earlier of whether or not we can diagnose this with all these other non-motor symptoms. And that's the exact problem, is it's associated with Parkinson's disease. And we think that we know some of the damage that might cause it, but it's also so common that unfortunately the only way still that we diagnose Parkinson's disease clinically is the presence of the cardinal motor symptoms. So there's a list that a neurologist will have to go down and say, do you have all of these things that meet the diagnosis? And those frequently will include bradykinesia, slowness of movement, tremor, and rigidity. Not all patients will present the same. And so you can have some, but not others. And the big addition to this list within the last 5 years was, if you do imaging and show a loss of sympathetic innervation in the heart, it's a supportive criterion for diagnosis. But it's not all that common for a patient to come into the neurologist, meet all the other criteria for Parkinson's disease, and then say, oh, also, can we just image my heart? That's sort of, it just feels sort of extraneous once you have this diagnosis. So it is a supportive criterion, but it's simply just not done as often clinically.
So I usually think of neurology and neuroscience as being with the brain. Or is this also neurology in the heart, even though it's not, it's a different organ?
Yes, though it will not get you as much attention at neuroscience conferences. I can tell
Wait
you
a
that.
minute, wait a minute. I'm detecting. I'm detecting.
Yeah, it is. This is the peripheral nervous system. And so the peripheral nervous system consists of sympathetic, parasympathetic, and super interestingly, if anyone here is into the GI tract, the enteric nervous system. It's its own little nervous system. It functions, it does get feedback from the parasympathetic and sympathetic nervous system and the central nervous system, but it functions semi-autonomously. It has the same diversity of neurotransmitters in the enteric nervous system as in the brain. The enteric nervous system is really cool. But yeah, it is all neuroscience. It's just that it does get a lot less attention and there is less research focused on it for a variety of reasons than the central nervous system.
And do we have researchers working on the digestive system that you had shown up there and all those problems like you are with the heart?
Yeah, so my first paper in grad school was actually on whether or not this animal model affected the enteric nervous system. So we looked at the neurons in the colon and found that this same neurotoxin, 6-hydroxydopamine, that causes a loss of sympathetic innervation in the heart, also causes a loss of neurons in the GI tract that produce a type of neurotransmitter called a catecholamine. So that's norepinephrine, epinephrine, and dopamine. So yeah, there are people doing research on it, and it sounds like you had someone who does swallowing research. That could have been Corinne Jones from my lab a few years ago, or there's a few different people on campus that do swallowing research in Parkinson's disease. So that would also be part of the enteric nervous system, from mouth all the way to the end of your to the end of your gut. So that's the esophagus is affected a little bit differently than the stomach and the colon. But yeah, there are lots of people doing research on it. Again, I think that there's a lot less clinical research on it than with the motor symptoms. I think mostly due to the fact that the motor symptoms make it very, very, very difficult to even function when they're at their worst. And constipation is extremely unpleasant, but is not as debilitating as the motor symptoms of Parkinson's disease when they're untreated and when they're at their worst. And so there's been a lot of focus on trying to improve motor symptoms so that Parkinson's disease patients can go about the rest of their life. Whereas, losing your sense of smell or constipation are unfortunate and sometimes worse than unfortunate. Sometimes they can really affect quality of life, but they don't tend to be quite as debilitating as the motor symptoms. And so that's really where the research has focused. And things, I think earlier I heard people talking about deep brain stimulation. Of course, the common treatment is oral dopamine replacement or levodopa. So those would be more the focus. And then lots of physical and occupational therapy is also very common for patients with Parkinson's disease.
I would think having more of the research on the heart or the gut would help with early diagnosis.
I think so. I think the tricky part there is how specific are our findings? And that will take more research, which of course, as a researcher, I heavily advocate for. But, to answer the question of if we find, what is the signature of Parkinson's disease in the heart? Is it a certain ratio of inflammation to oxidative stress? Is it inflammation, oxidative stress in the heart, plus a blood marker of this specific RNA? And so that's another part of my research that I did is what do we find in the blood? Is there something specific? Can we pair multiple things together so that we can get an earlier diagnosis? And that's something they've been trying to research clinically for a long time.
Well, even if it isn't Parkinson's, if there's damage to your heart that's causing fatigue, isn't that worth treating?
Yes. Yeah. And I agree with you on that. I think moving some of these radioligands into a clinic where we learn more about mechanisms of nerve damage in the heart, I think that has a lot of value. Potentially outside of just Parkinson's disease, but then we also have, frequently very common people will go to clinics for heart attacks, right, for myocardial infarction. So what can we learn about that? Interestingly, after a heart attack, rather than in the area where the heart attack was at its worst, and cells actually die, you'll have a loss of innervation, but right around that, you interestingly have hyper reinnervation of sympathetic innervation. So patients that have heart attacks will sometimes have too much sympathetic innervation right around that damage, and that can cause arrhythmias. So is there inflammation and oxidative stress associated with that? If so, where is it? Can we intervene? Would it make it better? I think those are all relevant questions. Yeah, I certainly, I mean, I have personal interest in it, but I certainly would be interested in knowing what these imaging techniques find in a clinical population in the heart.
One other question I have, is anybody looking at, like, you're giving one dose of a neurotoxin,
Yeah,
more of a chronic
yes.
dosage? And
Yeah.
whether there's any recovery after a chronic exposure?
Yep, so for effects in the heart, we are really the only group modeling in non-human primates this effect on the heart. So this is pretty much the model that exists right now. However, for motor symptoms, yes, for motor symptoms, people have been developing various different models of the effects on the brain in Parkinson's disease for quite a long time now, with a number of different neurotoxins. So as I mentioned, there are some people who try to model something that's a little more similar to what a patient might be exposed to, like rotenone or paraquat. That's been tried. It has some challenges though, because it's very, very hard on the animals. While it can produce the Parkinson's-like pathology, it also produces a lot of other problems for the animals, and we do need to take care of our animals. And so we need to be conscious of producing the pathology we need and not lots of other damage that is just going to make animals suffer. But there are people who do more chronic dosing. So for example, that neurotoxin that I mentioned, MPTP, where we tried it in the brain to test pioglitazone, that neurotoxin, if you give it peripherally, if you give it just like intramuscularly, it will cross the blood-brain barrier. So the toxin that I gave, 6-hydroxydopamine, does not cross the blood-brain barrier. So the monkeys that I worked with had none of the motor symptoms. But MPTP, if you give it intramuscularly, it will affect both the peripheral and central nervous system. And so there are some researchers that will give small doses over time, and with the rationale that then maybe you're reproducing some of the damage both in the brain and outside of the brain. Again, we have to be very responsible with how we create those models, because it is affecting the entire body. So that's actually one of the reasons that our group gives MPTP intracarotid and only to one side, is so that those monkeys then have full function of the other side of their body. So on one side, they have Parkinsonism, but on the other side, they're able to take care of themselves, to move around their cage, to complete their everyday activities. So yes, in short, that does exist, but it comes with a lot of challenges, including animal care type of challenges. But there are some people that do that. And then the goal with that model is then to stop sort of at what would be early Parkinsonism, when the animals start to have some effect on their gait. And then they'll do research on therapeutics, on what impact that has in different parts of the body. Yeah.
Can you go through the 4 grants that are listed there, what those are, how
Yes.
you're involved with those? You know them?
We'll start, this one is a fellowship that I received to fund the blood research part of my thesis work. This is through the NIH, through the National Heart, Lung, and Blood Association. This one, these two are both primate center, and I can't recall which one does which thing at the primate
The
center.
first one's the base grant.
Right.
Yeah, the second one I think is like a supplemental something.
There's one's based.
But yeah, this one is our base grant that funds our primate center. So Giordano probably covered this, but there are multiple national primate research centers throughout the country. And this is the funding for our primate research center here on campus. And this last one is an R21. So this is a grant that my PI received right when I started, beautiful timing, to essentially do the first half of this study. So this was the funding that allowed us to do all the PET scans. And so I came in right when this project was starting to do these PET scans. And then at the end, we added in the work looking at, we're actually looking at non-coding RNAs circulating in the blood with the F31.
So your lab, which is really interesting, and all of your research and everyone else's research projects that are under Dr. Marina Edwards' lab are actually part of UW-Madison's preclinical Parkinson's research program for the whole university.
Yeah. Yeah, so we try to, as you can tell, we try very hard to keep our research very clinically focused. So we spend a lot of time talking to neurologists and going to patient outreach groups. And I think there is a place for basic research. Basic research is extremely important. Someone has to sit down and figure out what protein does what in the body. But what we're more excited about is once other people do all the hard work of all the basic research, how can we take that and do something with it that potentially could be next in line for a clinical translation?
Your bridge.
Yes, yeah, a bridge between, the people working really hard with all their cells and mice and fruit flies and zebrafish and all the cool basic research models.
How about the stem cells? I know that they've gotten to the point where they are able to activate some cardiac cells, stem cell things. Now, wouldn't that then be able to be injected, be immersed, and bring back those cells in the monkeys that you have advanced down the road?
That is a really interesting question. So most of that research is focused on replacing the muscle cells of the heart. So most of that research is focused.
As opposed to neurons.
Correct. And there's sort of two reasons for that. The first is that heart attacks are much more common and much more deadly. And so that is simply where the clinical focus is. And so there's a lot of research now on injecting stem-like cells that are sort of positioned to become cardiomyocytes, the muscle cells of the heart, and injecting them where damage is in the heart. The second reason is that if I fly all the way back here, this is just a cartoon, so it's not entirely accurate. But the location of the actual cell bodies of the neurons that die are back along the spinal cord. So what you're seeing here is basically you have ganglia groups of neuronal cell bodies that run along the spinal cord, and they send out these really long axons that come all the way to the heart. And so if we wanted to replace these neurons, one, we'd have to put them back here, and two, we'd somehow have to talk them into coming all the way down here without going everywhere else all over the place. So that becomes very challenging. The reason that using stem cells to repair neuronal circuits in the brain is more effective is, one, it's just practical, it's more compact. And two, we can sort of, typically what's done, interestingly, although the cells in Parkinson's disease that die are these cells, and down here in the nigra, which would be down here, typically actually what's done in all that, there's ongoing clinical trials to try to replace these cells in the brain, but they're not actually usually put down here. They're actually usually put up in a part of the brain called the striatum, because this substantia nigra down here produces dopamine and it sends out axons from here to the striatum. And so most of the clinical trials that are going on right now have basically said, there have been lots of research, lots and lots and lots of research done on this in animal models, including lots and lots and lots in monkeys. And basically what the research shows is don't put it here, because then you have to somehow coax them into communicating here and not everywhere else. Put them right here. So you put them in the area where they usually are sending their signal, and then they're there basically being little dopamine factories. And so they're in a slightly different place than usual, but they're getting the dopamine to where it needs to go. So in the case of replacing cells in the brain, we can sort of skip a step in the circuit and put them right where they need to be. For replacing nerve fibers in the heart, it'd be a lot more challenging. Yeah.
What happens when you have a heart transplant to these
We have a
two?
question back here.
Oh,
too.
I'm sorry, go ahead.
Trying to get-
Oh, yeah, I was wondering, like all the progressive degeneration that's associated with Parkinson's disease, seems like most of the treatments and therapies are designed just to stop it where it is, but not actually reverse it.
Yeah.
I was wondering if any of this the cell replacement therapy has the potential to actually reverse some of the symptoms.
That's essentially the goal. Yeah,
Okay.
so as you can imagine, if you put the cells here and then they produce dopamine that's lost because these cells are dead, you would repair this part of the circuit.
Okay.
Now, one thing to keep in mind is you don't repair any of this, right? So really what you're doing is you're repairing the Parkinsonism, right? The Parkinsonism that's just these motor symptoms. But yeah, that's the basic idea. Is sort of pioglitazone. One of the reasons that stopping inflammation, oxidative stress might not be effective is because these cells are already dead. There's not a lot to save there. You can slow the progression, of course. It is a progressive disease, but you already have Parkinson's disease. Putting the cells here, what you're hoping to do is replace what's been killed, what's died, I guess, been killed. Very active term, but these have sort of died off their neurodegeneration. And so the idea, is to replace that. So then hopefully you get actual repair of the circuit. And so then patients would not just halt the degeneration, but would actually have some improvement. And then you had a question about a heart transplant.
Yeah, if you
Yeah.
go back to that slide and just show.
Yeah.
Liver is transplant pretty common, I think, heart transplants. What insights, if any, do you get about how these things work by studying transplants?
Yeah, so transplants, to my knowledge, there is very minimal regrowth of the autonomic innervation of the heart. There is some, but I don't think it's incredibly extensive. And so really what you're looking at is a loss of this. So as you can imagine, you'll be okay. You still have the heart functioning and moving blood like it needs to, in the same way that Parkinson's patients without this are okay. It's just that you have to be very careful because you're going to lose some of your body's natural ability to regulate your cardiovascular system. You're going to lose a lot of that autonomic feedback because you've really just sliced through that. But what's more important is that you have a functioning heart. So trade-offs.
Good point. Okay, it's just that exchange sometimes it's interesting to see what things happen and what things don't happen.
Yes, lots of downstream consequences once you start taking things out and putting things in the body, because yeah, there is an extensive nervous system outside of your central nervous system. Most people think of the brain and the spinal cord, but you know, everything that you do all the time is because nerves are sending signals to do it. And even the things you're not thinking about, right? Your digestion has sympathetic and parasympathetic input. So as I mentioned, if you were to cut off all the sympathetic and parasympathetic input to your GI tract, it does still have its own little enteric nervous system, but then you're going to lose rest and digest. So you're going to lose your body basically telling you, hey, you seem calm right now. Why don't you digest some food while we're waiting? You're going to sort of lose that, or you're going to lose the something's happening. It's terrifying. Let's not digest food right now. Let's focus on the scary thing. You're going to lose the fight or flight. So it will still function. It's just that sort of balance that we all kind of take for granted that's happening without us thinking about it is taken away.
More questions? Or are we done?